Assalamu alaikum, everyone. My name is Reena NoorEddin, and I'm a third-year medical student. I'm extremely excited to be teaching you guys your 4 physiology lectures of your MSK course. Now, this is my first time doing a recorded session, so I don't have anyone to let me know if I'm moving too fast or if anybody wants me to re-explain a concept. So if there's anything that you want me to re-explain, please feel free to contact me with my contact info mentioned at the end of the lecture. And before we begin, a good thing about these 4 lectures is that you already took almost 50% of the content, and the other 50% is actually really easy to understand. It's just that the terminology is a bit tricky. And without further ado, let's take a look at what we will be talking about today. Today, we're going to be discussing neuromuscular transmission, which is essentially how your body turns a nerve signal into an actual physical contraction. Then we will be going over excitation-contraction coupling, which is something that we took in foundation. Then we will be taking a look at the role of ATP in muscles and where exactly do we get this ATP from. Then we'll look at muscle contraction mechanics and the force regulation. And these 2 topics are going to involve some graphs. And then finally, we're going to take a look at smooth muscle contraction, and we will be comparing it with cardiac and skeletal muscle contraction as we are going through it. And without further ado, let's just begin with the lecture. So first thing we're going to be looking at is how exactly does our body turn a nerve signal into a physical muscle contraction. First things first, your skeletal muscle fibers are innervated by something called an alpha motor neuron. Now, your body has different types of neurons, and you'll be introduced to what each type has in second year. What you need to know for now is that skeletal muscles are specifically innervated by alpha motor neurons. Now, this motor neuron is going to go and reach the muscle fiber. And this region where the neuron reached the muscle fiber is called a neuromuscular junction. The neuromuscular junction is made up of 3 different components. The first one is called the nerve terminal, and that is your presynaptic terminal. So obviously, where the nerve is reaching the muscle, that is your presynaptic terminal, otherwise known as your nerve terminal. Then, in between the nerve and the muscle, you're going to have something called a synaptic cleft. And this is the gap between your nerve and your muscle fiber. So they're not directly touching each other. There's a small gap between the 2. Finally, you'll have something called the motor end plate, which is the surface that is closest to the nerve. And this is also known as the postsynaptic terminal. So once again, your neuromuscular junction is made up of the presynaptic terminal, also known as the nerve terminal, your synaptic cleft, the space in between the 2, and then your motor end plate, which is also your postsynaptic terminal, which is at the level of the muscle fiber. Now, what exactly happens at the neuromuscular junction? So once again, the way we're starting this story is let's say you put your hand over a stove, right? So your brain is sending a signal to your muscle that you need to move away. And this signal takes the form of an action potential. And we took this in foundation. We took what an action potential is in foundation. This action potential is going to go across the entire neuron like this until it reaches the presynaptic terminal over here. Okay? And essentially what an action potential does is that it depolarizes the presynaptic membrane. So if you take a look at the diagram over here, we can see that we have positive charges going down and across the presynaptic membrane. Obviously, that is because of the depolarization. And that is the first step. Now, what does this depolarization do? This depolarization is going to affect the calcium channels, specifically the voltage-gated calcium channels at the level of the presynaptic membrane. So essentially, your normal resting potential is -70 MV. And then what the action potential is trying to do is it's trying to get you to reach +30 MV. That's why we have the positive charge over here. Now, along the way, of course, as you're going from -70 and you're becoming more positive until you reach +30, you are going to reach the number -40. At this level, you're going to open the voltage-gated calcium channels at the level of the presynaptic membrane. And of course, once they're opened up, you have more calcium outside of the cell than you do inside of the cell. So calcium is going to rush in. So let's go over this one more time. Because of the depolarization, you're going to open voltage-gated calcium channels, specifically at the level of -40 MV. Once this opens, you'll have calcium rushing inside the cell. Now, a general rule in your human body is that calcium, once it's inside the cell, is most probably going to bind to some form of vesicle or compartment to get it to release its content. This is why your body fights so hard to keep calcium, very tightly regulated, because it can affect many cellular processes. So what's going to happen here is that this calcium will bind to this presynaptic vesicle over here, and it's going to cause it to fuse with the membrane and to release its content. Now, what does this vesicle contain? It contains a molecule called acetylcholine, or ACH. Okay, so once this calcium comes in, binds to the vesicle, and gets it to release its content by fusing with the membrane, this acetylcholine will end up going and binding to a receptor on the postsynaptic membrane. Okay, and the receptor is called the nicotinic receptor, but we will get into all of these details later. So before we move on, let's just take one quick recap of what I've just explained. First of all, you'll have a nerve signal going down the nerve, and this nerve signal takes in the shape of an action potential. Action potential means that you're depolarizing your membrane, or you're making it more positive. This depolarization is going to make you open your voltage-gated calcium channels, specifically at the level of -40 MV. Once these open, calcium comes inside the cell, it binds to the vesicle. Once it binds to the vesicle, it will fuse with the membrane, and now you have released acetylcholine. Acetylcholine from the synaptic cleft, it just floats around, and then it will bind to the nicotinic receptor at the postsynaptic membrane. And this is the first part of our lecture. Let's move on to exactly how does the calcium cause the vesicles to fuse together. And to understand this, we're going to have to take a look at the molecular composition of the synaptic vesicle and of the plasma membrane. So the synaptic vesicle, on its membrane, it has two very important proteins, the synaptobrevin and the synaptotagmin. Okay? What the synaptobrevin will do is that it will interact with two proteins found at the level of the plasma membrane. What are these proteins called? They're called syntaxin and SNAP25. It's very important for you to know which proteins are found on which membrane. So again, synaptobrevin and synaptotagmin are on the synaptic vesicle, the presynaptic vesicle, the one with the ACH. And on the plasma membrane, you'll have syntaxin and SNAP25. So the synaptobrevin specifically will begin entangling itself, as you can see over here, with the syntaxin and SNAP25 complex. So it's basically trying to bring the vesicle closer to the plasma membrane. However, there is something stopping it from completely fusing with the membrane just yet. And of course, that's going to be your synaptotagmin. As you can see, your synaptotagmin over here is kind of holding it back. It's preventing it from fusing. Why? That is because the synaptotagmin is waiting for the calcium to come in and bind to it. So once the calcium channels open and they allow calcium inside the cell, the calcium will come and bind to the synaptotagmin. This will cause the synaptotagmin to go, to move away. And now the synaptobrevin will successfully fuse with the syntaxin and the SNAP25, and this will cause the vesicle to fuse with the membrane. And this is exactly what I have written over here. Now, this fusion is going to require ATP hydrolysis, and it's facilitated by an enzyme called NCF. Don't forget that, okay? Now, moving on, once the ACH is found in the synaptic cleft, it is going to bind to the nicotinic ACH receptors found on the postsynaptic membrane. And this receptor is a cation receptor, meaning it allows positively charged molecules to go through, which is both Na and K, okay? So sodium and potassium. And the reason why we call it a nicotinic ACH receptor is because from the name, both acetylcholine and nicotine are going to interact with this receptor in a similar way. Now, one more important thing you need to know from this slide is that you need two ACH molecules to open this cation receptor. And this is really important. It came in an SAQ for our batch. You need two ACH molecules to open the nicotinic ACH receptor. Now, what happens after you open this cation channel? This concept is going to require us to go back to a concept that we took in foundation. So in skeletal muscles, your membrane potential is actually negative 90 MV. It's around that range. Okay, usually like in neurons and in the cells that we talk about when we're talking about action potential, our membrane potential is usually negative 70 MV. However, for skeletal muscle specifically, it's actually negative 90 MV. And the equilibrium potential of sodium is positive 30 MV. What does that mean? That means that if we open this channel, we're going to want to move the membrane potential closer to the sodium's equilibrium potential. So the inside of the cell is negative 90, and for us to make the concentration of sodium inside the cell and outside of the cell equal, we're going to want to reach positive 30 MV. Okay, I hope that was clear. So for us to jump from negative 90 to positive 30, that is a very huge jump. And essentially what that's going to do is you're going to end up having a lot of sodium rushing inside the cell to get this negative 90 from inside the cell closer to the positive 30 that is the equilibrium potential of sodium. So to recap, what I'm trying to say is that when you open the acetylcholine channel, you're going to have a lot of sodium rushing inside the cell because of the driving force. If you understand that much, then that is enough. If we take a look at potassium's equilibrium potential is actually also negative 90. So they're not exactly equal. So this is just a rough estimate of the numbers. It's the membrane potential of a skeletal muscle is almost 90, and the equilibrium potential of potassium is also almost 90. Point is, you're going to have a very, very weak outward flow, okay? So you're going to have a few amounts of potassium ions leaving the cell. So if you take the amount of sodium entering the cell and the amount of potassium leaving the cell, overall, the point is you're going to have a positive charge on the inside. Why? Because you have so much more positive charge entering the cell than the amount of positive charge leaving the cell. So once again, what I want you to take away from that entire explanation is that more sodium will enter than potassium leaving. And this will cause what to happen? This will cause a depolarization to occur. However, this depolarization is not an action potential. It is something that we call a graded potential, or specifically in the context of skeletal muscles, it is also known as an end plate potential. Now, what does end plate potential or graded potential mean? I do think you're familiar with graded potential from a previous lecture, but nonetheless, a graded potential is a weak temporary change in the membrane's charge, okay? So you're very temporarily going to change the membrane's charge, and it's going to depolarize a little. If this depolarization is strong enough to cross a certain threshold, then you're going to trigger an actual action potential. Okay, so as we said earlier, the skeletal muscle potential is negative 90 MV. And the threshold for skeletal muscle to trigger an actual action potential is negative 75 MV. This is like a 15 MV difference, okay? So what's going to happen is that this nicotinic channel, the one that we just discussed, is going to allow this much sodium, a lot of sodium to come in, which is going to make you have a positive charge. If this temporary positive charge, which is a graded potential, not an action potential yet, if this difference is more than 15 MV and you cross the negative 75 millivolt threshold, then you're going to trigger an actual action potential. And over here, I just wanted to remind you that graded potentials, they will decrease with distance, which is why I was saying that they were temporary, because as they spread throughout the cell, they're going to lose their momentum, which is why it's important that they're strong enough to overcome the threshold. And that is the main takeaway of the slide. So now what would happen if your graded potential or your end plate potential in the context of skeletal muscles, what would happen if it's not big enough to trigger an action potential? In those cases, we call this a miniature end plate potential, an MEPPS. And they usually occur spontaneously, like for no reason, or because of a drug called D-tubocurarine. And then the doctor put the slides, he just put graphs to, illustrate what this process looks like to you. So normally, you're going to have the nicotinic channels opening, and this will trigger a graded potential, which is over here. And that is going to overcome the threshold, triggering an actual action potential. That's all you need to know. Now, you can see over here that the numbers don't line up with what I explained, but I triple checked the numbers that I gave you over here, which is the negative 75 and the 15 MV. So I'm pretty sure that this is just an issue with the graph, okay? Now, as you can see with D-tuberculine, which is the drug that usually causes miniature end plate potentials, what happens is that the graded potential is too low for it to cross this threshold. Therefore, you're not triggering an action potential the same way you do normally. Now we're going to move on to something called the quantum release concept. So in one vesicle, you're going to have almost 10,000 ACH molecules. And there's a theory that says one vesicle is going to equal to one quantum. And then this theory also says that one quantum will be enough to elicit a 1 MV of depolarization. So following what we were explaining earlier in this slide, which is that we need a 15 MV difference, the theory basically says that you're going to need 15 quantums at least if you want to trigger an actual action potential. And then it goes on to say that in a normal physiological nerve action potential, you usually trigger 100 vesicles. So we were saying that we only need 15 to trigger an action potential, but normally your body releases almost 100 vesicles. Therefore, your body will usually be able to produce action potentials very, very easily, unless you're taking a drug or you have some sort of disorder. That is what the slide is trying to say. It just tries to put numbers to everything that you've learned. Now, this slide just has some more general rules, starting with that obviously each skeletal muscle fiber will extend from one tendon to the other, and then the nerve will just kind of come in the middle of this muscle fiber, and the depolarization will occur in both directions, like this. It's really important to note that we do not spread the action potential from one muscle fiber to the next. We do not have gap junctions along the two, allowing the spread of electricity between skeletal muscles. Every skeletal muscle fiber has to be innervated by a neuron. Now we'll be moving on to vesicle recycling, because if you're imagining it, if we keep fusing vesicles with the membrane so that we're releasing the ACH, eventually you're going to end up with a really, really long presynaptic membrane because you just keep adding vesicles to it. So your body has to regulate and it has to recycle those vesicles. How does it do that? It needs a molecule called clathrin. And this clathrin molecule has three important subunits that help facilitate this process. First one is called adaptin. So what adaptin will do is that it will move, it's all a part of clathrin. So the adaptin part of clathrin will move along the presynaptic membrane, and it will recognize that this portion looks like it came from a vesicle, and it will mark it. It will just stand over it and mark it. So what does adaptin of clathrin do? It will recognize this portion of the presynaptic membrane is actually from a vesicle, and it will bind to it to mark it. Then the next portion of clathrin, called the triskelion, is going to, it's like a triangle shape, like this. So it's going to pinch the vesicle membrane and make it look like a ball again. And then the final part of the clathrin, called dynamin, is going to use GTP to pinch the vesicle away from the membrane again and to make it look like this inside the neuron. So let's take a look at this slide where I wrote everything out for you. Number one, adaptin, which is a part of clathrin, is going to recognize that this area looks like it came from a vesicle and it will bind to it. Then the next part of clathrin, called triskelion, which is this triangle shape over here, is going to pinch it back into looking like a vesicle. Then dynamin, by using GTP for energy, is going to cut it off from the membrane, returning it into this formation. Then to lose the clathrin from the entire vesicle, you're going to use ATP. So there are two steps that need energy. Number one is dynamin to pinch it off. Number two, you're going to need to release ATP for you to release the clathrin. And now you have an uncoated vesicle that's ready to be reused. All of these uncoated vesicles will bind together and fuse together to form what we call an endosome. And then this endosome, on its surface, it has receptors. And through these receptors, you don't need to know the name, but through these receptors, ACH will come back in, and you can just continue the cycle from here. Now we understand how to recycle the vesicles. What about the acetylcholine that we put inside the synaptic cleft? Okay, we know that it binds to the receptors over here. After it's done binding to the receptor, what does it do? Where does it go? You have an enzyme found in the synaptic cleft called acetylcholine esterase. And what this acetylcholine esterase is going to do is that it's going to break down the acetylcholine into choline, which is recyclable, and other waste products that we don't need to discuss, they're just going to be discarded. But the choline is pretty important because we're going to recycle it back into our cycle. So the choline will go back from the synaptic cleft into the neuron, and inside the presynaptic terminal, it is going to be bound with newly formed acetyl coenzyme A through an enzyme called choline acetyltransferase, and it's going to make new acetylcholine. So again, what's important to note is that choline is recycled, acetyl coenzyme A is not recycled. The acetylcholine esterase is found floating in the synaptic cleft, and the choline acetyltransferase is found inside the presynaptic terminal, and it helps combine choline and acetyl coenzyme A. Now let's talk about the clinical applications of everything we've learned. Mainly, the way we use the neuromuscular junction in medicine is that we try to block it because we want to avoid contractions in certain situations, specifically in surgery. So we use neuromuscular junction blockers during surgery because we don't want our patient to have contractions randomly throughout the surgery because that would disrupt the surgery. And the two main types of blockers, because over here the doctor put a bunch of groups, and I would suggest that you study them anyways, just in case you get asked a low yield question, but I did outline what the high yields are. So the two main types of blockers that you need to know are something we call acetylcholine antagonist and acetylcholine agonist. And the way they work is kind of similar, but it's also different. So what the antagonists will do is that they will go to the acetylcholine receptors, the nicotinic receptors at the postsynaptic membrane, and they will bind to them over here. And when they bind to them, their function is essentially to block the actual acetylcholine from coming and performing its function. So again, your antagonists like D-tubocurarine and bungarotoxin are going to go and bind to the ACH receptors and block the actual ACH from coming in and opening up the channel. It will just come and it will sit in the place of ACH and it will not trigger any reaction. So obviously, because these channels are not opening, you will not have an end plate potential. And if there's no end plate potential, there's no action potential, and that means there's no contraction. Now, what do your agonists do? They work in the same way as antagonists in the sense of the agonists will also go and bind to the receptor. However, they're different because unlike the antagonists, they will actually trigger a small response. How? Let's take succinylcholine. Succinylcholine will go and it will also bind to the receptor. But what succinylcholine does is that it will open up the receptor, just like ACH will do. It will open it up, leading to initial tremors. So when you give the patient succinylcholine, they will initially have tremors. What it does instead of acetylcholine is that it will keep the channel open for a very, very long time. So usually acetylcholine just comes and binds, it opens the channel, you'll have, the whole contraction and everything. And then after that, the acetylcholine esterase is going to go and destroy the acetylcholine, so you will relax. What happens with succinylcholine is that it will go and it will bind to the receptor, it will open the channel, causing tremors at first, but then it will keep the channel open. And you basically just don't get a chance to relax and to start having contraction again. So what will happen to these receptors is that they will enter a refractory state. They'll just stop working after a while. And what happens there is that you'll end up with paralysis. So these drugs we mainly use in surgery, as I said, to induce paralysis because you don't want contractions happening throughout the surgery. Now, another important toxin, not a drug, it's a toxin, that you probably heard about before. If you guys remember, we had an outbreak, I think last year, around this time, is the botulinum toxin. So what botulinum does is that it will go to the vesicles, the presynaptic vesicles, and it will destroy their proteins. So without their proteins, obviously, they cannot do the whole fusion anymore. And without fusing with the membrane, they will not be able to release the ACH, which means you will just not have any contraction and you will just have paralysis. And that is how botulinum works. Now, what about tetanus? I don't know if you guys heard of tetanus before, but the way tetanus works is, for example, if somebody gets scratched by a rusty nail, you usually have to rush them to the ER so they can get a tetanus shot. Because what happens with tetanus is that you'll end up with very severe spasms of your muscle. And how does this happen? So the entire lecture, I've been talking about how neurons will simulate muscles to contract, but just like how you have neurons to stimulate contractions, you also have neurons in your body that inhibit contraction. And tetanus actually works on the inhibitory nerves. So it's going to work in the same way as botulinum by going to the presynaptic vesicle, and it's going to inhibit its fusion, but it's working on the inhibitory nerves and not the stimulatory nerves, and that's going to cause an uncontrolled contraction instead. So if you just understand what I wrote over here in the table, that's enough, because there's a lot of new terminology to be introduced with tetanus. Now, the doctor also introduced an autoimmune disease called myasthenia gravis. And the way this disease works is that you have antibodies against the ACH receptors over here in the postsynaptic terminal. These antibodies will attack the receptors and they will destroy them. And because you have fewer receptors, that means you're going to have weaker contractions because obviously you're not causing the same physiological changes anymore. Finally, you have another autoimmune disease called Lambert-Eaton syndrome, and you do have more antibodies also, but these antibodies will act on a different receptor, specifically the calcium channels, which are found on the presynaptic membrane. They will attack these channels, destroy them, and because you don't have calcium entering and causing the fusion of the vesicle, you just won't have contraction. So the only one that will cause severe contractions is tetanus. Everything else I explained is going to cause paralysis, while keeping in mind that succinylcholine will cause initial muscle tremors before it causes paralysis. And with that, we have finished our first lecture. We will be continuing on to the second lecture. Just give me one moment, please. And let's get into our next lecture. So, now it's better if you studied your histology lecture before you got into all of your physio lectures, but if you haven't, that's fine. I will be teaching you everything that's relevant to the lecture anyways. But just so you can better understand the structures and what they're doing, it really is advisable that you just get your histo lectures out of the way first. So let's take a look at what a skeletal muscle cell looks like. Something that you'll need to appreciate about a skeletal muscle cell is that it's not like your typical cell where you have cytoplasm and like organelles freely floating around. It's actually a very densely packed structure. I'm not saying it doesn't have cytoplasm. I'm just saying that on the inside, it's not free cytoplasm and free organelles just roaming around. It's a very organized structure. And the reason why it's so organized is because of the actin and the myosin filaments, which are organized in those structures called myofibrils. Okay, so as you can see in the photo, we have a bunch of myofibrils stacked on top of each other, creating this sarcomere, which is what gives our skeletal muscle the striated appearance. And these myofibrils are wrapped by your plasma membrane, your sarcolemma. And this is how your skeletal muscles stay so organized. Look at how it wraps around it. Now, the sarcolemma of your skeletal muscles has a very special feature where the sarcolemma will actually invaginate into the cell. So as you can see over here, this is the sarcolemma, the plasma membrane, and then these tiny holes are the invaginations into the cell. You can see them make like a tube-looking structure inside the cell. Why does it need to invaginate like this? This is because the depolarization occurs at the level of the plasma membrane. And because we want the depolarization to reach our, contractile structures, our myofibrils, our actin and myosin, we want the plasma membrane to go inside and dip towards these structures. And that's exactly what a T-tubule is. A T-tubule is going to be the invaginations of the sarcolemma into the cell. And then around this T-tubule, this purple structure, around it, you can see the sarcoplasmic reticulum organized like this, which is pretty unique to muscle cells. So every T-tubule and two sarcoplasmic reticulums, so this formation over here is going to be called a triad, okay? And I essentially sum all of that up in this slide. In skeletal muscles, you have something called a triad. What's a triad? It's one T-tubule with two sarcoplasmic reticulums. What's a T-tubule? The T-tubule is just an extension of the sarcolemma. It is the inward extension of the cell membrane. Why does it need to extend into the cell? Because we want the action potential to penetrate into the myofibril so you can reach our filaments. Excuse me. And then we also have the sarcoplasmic reticulum. What does the sarcoplasmic reticulum do? It stores the calcium for us because calcium is very important for contraction. So altogether, what does this triad formation do? This triad formation is important because we're essentially trying to translate our action potential, our electric signal, into our physical contraction. And please note that everything I'm explaining from here on out is going to happen at the level of the postsynaptic membrane, so our muscle fiber. We're done with talking about our neurons, okay? So, at the level of the neuromuscular junction, last time we left off was that we finally triggered an action potential in our muscle fiber. This action potential is going to go across the sarcolemma, and then it's going to go down our T-tubule, okay? Once it goes down our T-tubule, it's actually going to reach a structure called a DHP receptor. If it sounds familiar, this is because we did mention it in a previous block. I think it was foundation or CVP. Now, in skeletal muscles, this DHP receptor is a sensor, and it's not a channel. And I'll come back to this later, okay? So the DHP receptor, this voltage sensor, is going to sense that, okay, we have depolarization going on. And what this DHP receptor is going to do is that it's going to tell the ryanodine receptors of the sarcoplasmic reticulum to open up, okay? So once again, you have action potential over here from the neuromuscular junction. Then it's going to go across the sarcolemma of the muscle fiber, and it's going to go down the T-tubule, as I said, because we want to reach our contractile apparatus. Once it goes down the T-tubule, it will reach the DHP. DHP is a sensor, and it's not an actual channel. It will sense the depolarization, and it will tell the ryanodine receptors to open up. Ryanodine receptors are found at the level of the sarcoplasmic reticulum. So what's stored inside the sarcoplasmic reticulum? Calcium. And now you'll have release of calcium from the sarcoplasmic reticulum into the cytosol. Now, because of the cytosolic calcium, this cytosolic calcium is actually going to bind to troponin, which will trigger our actin-myosin interaction, and that is how we start contraction. And we'll revisit this concept again. But first, let's talk about calcium inside our skeletal muscle cell. So, in all cells of your body really focuses on maintaining the right amount of calcium in the cytosol because, as I said, you can trigger a lot of cellular responses. In skeletal muscles, you can trigger unintentional contractions and lead to spasms and cramps. In other cells, you can trigger apoptosis. Point is, your cells are really sensitive to calcium. So in your cytosol, you usually have a very low concentration of calcium. However, in normal conditions, your extracellular calcium, so the amount of calcium outside of the cells in the extracellular fluid, is very high. It's 1.8 millimole. And your sarcoplasmic reticulum calcium, so the amount of calcium in your sarcoplasmic reticulum, is very, very high. It's almost 3. Now, let's go back to the DHP receptor. And the reason I'm bringing this up is because, as I mentioned earlier, you did take DHP receptors from a previous lecture in either foundation or CVP when we were discussing the cardiac muscle contraction. And the way that DHP works in skeletal muscle and in cardiac muscles is different. So once again, in skeletal muscles, the way it works is you get the depolarization at the level of the T tubule, and your DHP is going to sense it. Why? Because it's a voltage sensor. So once it senses it, it's going to tell the ryanodine receptor, you need to open up, and it causes a mechanical change. It just pulls it up, and now you have a mechanical change. And now you have your calcium inside the sarcoplasmic reticulum is going to escape. In cardiac muscles, on the other hand, let's take a look at the DHP here. You'll have the same exact pathway from the neuromuscular junction. You don't have a neuromuscular junction in cardiac cells, but point is, once your cardiac cell 2 depolarizes down the T tubule, it's going to reach a DHP receptor. However, the difference here is that your DHP receptor is actually a calcium channel. Take a look at the model. This is your DHP receptor over here. And once it detects the depolarization, it opens up a gateway. This gateway will allow calcium from outside of the cell to enter the cell and bind to the ryanodine receptor. So you get the difference now? In skeletal muscles, you detect the depolarization, and the DHP pulls the ryanodine up, allowing calcium to go out. It's different in cardiac muscles. You will get the depolarization, you will open up the DHP, calcium will go in from outside of the cell into the cell. It will bind to ryanodine receptors, and now the ryanodine receptors will open and cause calcium to leave the sarcoplasmic reticulum. This is called calcium-induced calcium release because you use calcium to release even more calcium. And that is the difference between skeletal and cardiac muscles. So over here, I had a question prepared in case this was going to be an interactive session, but this is a pre-recorded session. So I'll just give you guys a moment to just read and try to solve it on your own. Okay, so assuming you can pause the video, the answer in this case is actually A, because the question is saying in a calcium-free solution, which type of muscle will contract? Skeletal muscles do not need extracellular calcium to trigger a contraction. They get their entire calcium from their sarcoplasmic reticulum. Cardiac muscles, on the other hand, need to undergo calcium-induced calcium release. They need to have calcium in the extracellular fluid so that they can trigger their ryanodine receptors to open up and to release even more calcium. That is the idea behind the question. Now let's talk about the thin filaments inside our skeletal muscle. Thin filament, from before, thin filaments refers to actin. What is actin? What is your thin filament? It's a double-stranded helix of actin, and you have exactly 13 monomers per strand. Let's take a look at the diagram over here. We can see in the green, we have our actin monomers, and then you have a ribbon in orange called the tropomyosin. And then on the actin, you have something called the myosin binding site. Your tropomyosin is typically inhibiting the myosin binding site when you're at rest. And this is something very important because we'll come back to it in lecture number 4, okay? So your tropomyosin is typically inhibiting the contraction at rest by hiding the myosin binding site. Then you have something called a troponin complex, which is made of TNT, TNC, and TNI. TNT is kind of like an arm that is attached to the tropomyosin. So when we want to move the tropomyosin around, who helps with the movement? TNT. Now, TNC, on the other hand, is where our calcium from the sarcoplasmic reticulum is going to bind. On the TNC, you have 4 binding sites. 2 of them are high affinity, and they always have calcium bound to them, regardless of the time, okay? Regardless of whether you're about to contract or not, they always have calcium bound to them. The other 2, that is the place where the calcium released from the sarcoplasmic reticulum will bind to, okay? And then you have your TNI, which usually works when you're at rest. So when you're at rest, when you don't have anything bound to the TNC, just the 2 high affinity sites, when you don't have anything bound to the 2 low affinity sites of TNC, your TNI is just going to keep the actin tightly bound to itself so that you don't relax the actin and cause the tropomyosin to move away, revealing the myosin binding site. So again, let's go over the slide again. We have the tropomyosin, which is covering the myosin binding sites. we have the troponin complex made up of T and T, which is the arm that's bound to tropomyosin. And then we have T and C, which is going to bind it to calcium. And we have 4 sites for this, 2 high affinity sites, always occupied with calcium or magnesium, and 2 low affinity sites where the calcium from the sarcoplasmic reticulum is going to trigger a contraction. And finally, T and I, which usually works when we're at rest, is going to inhibit contraction. How? By binding to the actin and keeping the structure tight, okay? Now let's talk about our thick filaments. Our thick filaments are also known as myosin, and there are a hexamer. Hexamer meaning they're made up of 6 components. The heavy chain can be broken down into 3 parts, and this is what our heavy chain looks like. So the heavy chain is made up of the tail, this portion. Then it's made up of the hinge, which is the portion that moves around during cross-bridge cycling. And then we have the head over here. The head has 4 light chains, 2 alkali and 2 regulatory, and it's where we're going to bind to the actin. So the head specifically is what binds to the myosin binding site. And then finally, the head also has intrinsic ATPase activity. So ATP will bind to the head, and the head has the ability to break it down into ADP and P. And this is, and at rest, the myosin head will usually have ADP and P, phosphate, PI just means phosphate, bound to it, okay? And I'll explain where this ATP comes from when we get into cross-bridge cycling. So the 2 alkali light chains are going to stabilize the head, and the 2 regulatory light chains over here, these are going to act on the ATPase activity and regulate it. So they're, it's either going to increase activity or decrease activity based on phosphorylation. And we'll talk about this more in the smooth muscle lecture. Now, when you're at rest, as I said, when you're at rest, your T and I is going to hold your actin in place, not tropomyosin actually. So it's going to keep your actin tightly bound. And this is going to help ensure that the tropomyosin is blocking your binding site. Your myosin, on the other hand, has ADP and P. And you may be asking, where did this ADP and phosphate come from? We'll explain when we get into cross-bridge cycling. What I want you to just remember is that at rest, your myosin head has ADP and phosphate, which means they're ready to bind to the actin, but they physically cannot bind to the actin. Why? Because of troponin I. Troponin I is bound to actin, and it keeps the actin structure very tight. Because the actin structure is very tight, the tropomyosin will inhibit the myosin binding site. So how do we initiate cross-bridge cycling? After you get calcium released from the sarcoplasmic reticulum, it's going to go and bind to the 2 low affinity sites on troponin C. This will trigger a response. This will tell the troponin I that you need to relax and you need to loosen the actin. Troponin I won't be released, it will just loosen the actin. And this action of loosening the actin will cause the troponin T to move the tropomyosin. So I know this gets just a teensy bit confusing because there's a lot of visualization going on, but if you can just memorize that troponin I is bound to actin and troponin T is bound to tropomyosin, I think you'll get the necessary questions correct, okay? And once this shift in the tropomyosin occurs, you will reveal the myosin binding sites, and now the myosin head can bind to actin, and you can have cross-bridge cycling. So let's go over the steps of cross-bridge cycling again. So let's start with number 1, where we are at rest. At rest, troponin I, TNI, is going to hold the actin very tightly, and this is going to cause the tropomyosin to cover the myosin binding site. Your myosin head is going to have ADP and phosphate attached to it. When your myosin head has ADP and phosphate attached to it, this means that it's ready to bind to the actin. It's just that the actin has the tropomyosin covering the binding site, so you can't cause a cross-bridge cycle. Eventually, you're going to trigger a contraction by releasing calcium from your sarcoplasmic reticulum. So the whole cascade of action potential and then DHP, RYR, calcium release, that's all going to happen. And the calcium will go bind to T and C, causing T and I to loosen up, causing the T and T to move the tropomyosin. Now that the tropomyosin is out of the way, the myosin head will immediately bind to the actin. Now, once your myosin head binds to the actin, you're going to release this phosphate group. And it is this release of the phosphate group that triggers our power stroke. And this is important because it can come as an MCQ. What triggers the power stroke? The release of phosphate from the myosin head. Okay? And once your myosin heads once the phosphate is released and you have the power stroke, your myosin head is going to move and change its conformation. The filaments will slide past each other, and now you have contraction occurring. Once you're done with the contraction or you reach the length that you're trying to reach, what's going to happen is that you want to lock your myosin head in that position, okay? And the way you do that is by releasing ADP. So what locks the myosin head in that position? What will stop the sliding of the filaments? The release of ADP. So now in stage 4, we're locked, okay? And the myosin head cannot move and you cannot relax, not until you get ATP bound to the myosin head. And this is really important because when your body does not have enough ATP during contraction, you will end up with spasms, or you will end up in a state called the rigor state. And this also happens in people who are dead. In people who are dead, they don't have ATP, so they will enter a state that we call rigor mortis. And that means all of their muscles are tense because there's no ATP to bind to the myosin head and to release it. So under normal conditions, you are supposed to get the ATP, and the ATP will come in, bind to the myosin head. Once the ATP binds to the myosin head, it will let go. Once it let go, the myosin head, remember, has intrinsic ATPase activity. And what that will do is that it's going to cause hydrolysis. And now you're back to stage 1. You're at rest. You have ADP and P. The actin should have reset. Or if it didn't reset, you can just continue contractions all over again until we reach relaxation, which I'll talk about how we reach relaxation. And over here, I just wrote everything down for you guys. So you can go through that if you need, just to read through it again. I do want to remind you before we move on from this concept that if ATP is absent, it's really important to understand that you will not detach the myosin from the actin, which means you will enter a rigor state if you're alive, and if the patient's dead, then we call the state rigor mortis. Okay? And you need to have 2 important conditions for the cross-bridge cycle to continue on and on and on to continue contractions. 1, you need ATP, obviously. And number 2, you need calcium so that, you can continue having the TNC and the TNI and TNT working properly, and you could keep having the binding site exposed. Now let's talk about relaxation. So how did we trigger the contraction in skeletal muscles? We triggered it by releasing calcium from the sarcoplasmic reticulum. How will we trigger relaxation? We will trigger it through the exact same way, which is we need to bring back that calcium from outside of the sarcoplasmic reticulum back into the sarcoplasmic reticulum. So to enter relaxation, we need to decrease the amount of cytosolic calcium. And how will we do that? We will do that using the help of sarcoplasmic reticulum calcium ATPase pump, which is called SERCA. And we did take this before in the lecture that we took earlier this year. And what's going to happen is that your calcium will be pumped back inside the sarcoplasmic reticulum. And to do this, you're going to use ATP. So just remember that for this process, you need ATP. What happens inside the sarcoplasmic reticulum is that you will have a protein called calsequestrin, which will further bind the calcium. So inside the sarcoplasmic reticulum, you have calsequestrin, which will just store more calcium and keep things organized and just help the sarcoplasmic reticulum maintain such a high amount of calcium. Now let's compare this to cardiac muscles, because as I mentioned, we already did hear about SERCA, because cardiac muscles do have SERCA, and they work in the exact same way. SERCA is found at the level of the sarcoplasmic reticulum over here, and it's going to pump calcium back inside the sarcoplasmic reticulum, and it will use ATP. However, because cardiac muscles also get their calcium from outside of the cell, from the extracellular compartment, then you need to remember that some of this calcium needs to go back outside of the cell into the extracellular compartment. How will it do that? Through 2 important transporters. First one is the sodium calcium exchanger. If you guys remember, this was called the NCX transporter. So I think calcium out, 3 calcium in, You don't need to know that. You just need to know the sodium-calcium exchanger. And the other thing that you need to know is the calcium membrane ATPase. So it's over here. You kick calcium out, but you have to use ATP. So for cardiac muscles, you have circa, which is like the skeletal muscle, but unlike skeletal muscles, you also have something called the sodium-calcium exchanger, or the NCX, and you also have the plasma membrane calcium ATPase. Now let's talk about the sources of ATP in muscle, because throughout this entire lecture, I've mentioned multiple times where we need to use ATP. So the number one source of ATP is, well, actually, this is not the number one source, but our first source of ATP is called the free ATP in the cytoplasm. So from the name, it just means that you're going to have free ATP floating in your cytoplasm. The limitation of this source of ATP is that it's very limited. It can only support 1 to 3 contractions before depletion. So your body doesn't really depend on it to do anything. It's just there. Now, what about, okay, now we have another method called the creatine phosphate system. What the creatine phosphate system does is that it will use ADP, add it with creatine phosphate to make ATP and creatine. And this is the fastest source of ATP in your muscles. It can deliver 4 moles of ATP per minute. The issue with this system is that it's very limited, right? You have a limited supply of creatine phosphate inside your muscles, and once this creatine phosphate runs out, you can't really make more, especially because to make more creatine phosphate, you need ATP. So obviously, the whole reason why you're using this system is to make ATP. So for you to remake more creatine phosphate, you're just going to have to wait until you're at rest. However, this system is still very important for muscles that need very quick sources of ATP. Our second fastest method is anaerobic glycolysis. Please excuse. Okay, please excuse that. I had a slight interruption. Now, moving on, anaerobic glycolysis is the second fastest source of ATP in muscles. So I'm pretty sure you guys are familiar with anaerobic glycolysis from your previous lectures and from your biochemistry lectures in semester 1. You are turning glucose into lactic acid to make 2.5 moles of ATP. So it is slower than creatine phosphate, but it's still one of the faster ones in your body. The issue with this one is that it can produce lactic acid, which will lower your pH and contribute to muscle fatigue and cramping. Now, the final source of ATP in your body is aerobic metabolism. So that means you have two sources for this. Number one is glucose. So you get pyruvic acid from glucose, and then you enter the Krebs cycle and the ETC cycle to make ATP, or you use fatty acids and undergo oxidative phosphorylation to make ATP. Now, this is slower than the other types that we explained. However, it's unique because as long as you have oxygen, you can use this for as long as you need, and it's basically unlimited, which is important for certain types of muscles. So let's talk about these types of muscles that I've been referencing. The fast twitch muscles, also known as white muscle fibers, these rely more on creatine phosphate and anaerobic glycolysis. Why? Because from the name, they're fast twitch. You want instant sources of ATP, and these are not really used for long-term exercising or like long-term movements. You just need them instantly. Now, on the other hand, slow twitch muscles are muscles that you use throughout the day for long periods of time, like your posture, like the muscles that help you sit up straight. You're using that throughout the entire day. Imagine if these fatigued quickly. So these usually rely on aerobic metabolism because the contractions are slower, but your body has a high endurance to these. Your muscles need to have a high endurance to be able to perform their functions correctly. And with that, we're done with our second lecture. I was going to take a moment break here, but it's fine. We can just continue. Let's continue with introducing what isotonic and isometric contractions are. And to do so, I'm going to give a gym example. And if that doesn't make sense, we can just move on to the table that I have written after the slide. So imagine you're trying to perform a deadlift, which is this exercise over here, and you're trying to break your personal record. So this is your first time lifting a weight that's this heavy, okay? And what's essentially going to happen is that you're going to try to pull it up, and you're going to strain a lot, especially if this is your first time picking up such a heavy weight. Your muscles are going to strain a lot, and your muscles are going to increase the force production that they have until they try to reach the maximum force that they can produce, okay? So in this state, your muscles are trying to reach the maximum force they can produce. I'm going to write max force. The tension over here is increasing as much as you possibly can because you're trying your best to move this force that you haven't moved before. But it's really important to note that in this state, you are not moving the force just yet, okay? And because you're not moving the force, that means you yourself are not undergoing any movement. So there's no movement and there's no velocity, okay? This is referring to your isometric contraction, okay? In isometric, the word iso just means same. Metric means length, okay? What length are we talking about? We're talking about your muscle length. You're not performing any movement over here. So your muscles are not shortening, okay? But at the same time, your muscles are still contracting. In both isometric contraction and isotonic contractions, your muscles are contracting. However, in this case, you're not producing any movement. Your muscles are not shortening, so they're isometric. Instead, you're trying to maximize the force that you're producing. And your movement will be zero, your velocity will be zero. Let's say you finally manage to pick up the load, okay? This right over here, you are performing movement. So you move from isometric to isotonic, okay? And what isotonic means is that you're going to have the same tone in your muscles, as in the same tension, as in the same force. So once your body's like, okay, I finally managed to pick this up, it's going to hold that force, it's going to hold that tension in its muscles until you can do the movement that you're trying to do. Like imagine with bicep curls or whatever, when you're doing a bicep curl, your muscles are contracting and they're shortening, but you're still maintaining the same tones in your muscle as you're doing those movements. Then once you stand up, like once you're done moving from the squat position to standing up, your muscles are done shortening. So you're done with the isotonic contractions and you go back to the isometric contraction as you're trying to stay holding it up. Let's go take a look at this table. We have isometric, as I explained, same length, meaning you have a constant length, your muscles don't shorten. Instead, you reach what's called an F max, maximum force. Why do you need to reach F max? Well, there's no reason why you need to reach F max, but the point of isometric contraction is that you're trying to reach F max. You're trying to maximize the force or the tension in your muscles. You will have no movement, which means you will have no velocity. And examples are when you're pushing against the wall or when you're holding a heavy object in mid-air, you're holding it steady, as you're like in this position for the deadlift. Okay? But when you're lifting the weight up, when you're going from this position to this position, that is isotonic contraction because your tension will stay the same. Your body's like, okay, this amount of force can move this object that I'm trying to move. So let's hold that force as much as we can. It will maintain the same tension, and your muscles will shorten to produce the movement that you're trying to produce. So yes, there is movement. Yes, the velocity is over zero. Doesn't matter what the value is, it's just over zero. And finally, the examples are like when you're lifting away and walking and running. Now we'll be taking a look at factors that influence the relationship between your muscle fiber length and the active force it produces. And we mainly have two. First one is the overlap between thick and thin filaments, so in your sarcomere. And the second one is calcium sensitivity, but this mainly applies to your cardiac muscle. So let's take a look over here. Over here, we can see, actually, let's leave this slide to later. Let's go down to discuss your muscle composition. Your muscle composition is made up of two very important structures that contribute to the overall tension. These two types of structures is either contractile proteins, like your actin and your myosin, or connective tissue elements like your collagen, your titin, your elastin. So the connective tissue elements, specifically the ones that are wrapped around your muscles, or the actin and the myosin, which are found inside your muscles, okay? The contractile proteins are going to generate active tension. What does that mean? That means they're going to generate active tension using ATP for force production. On the other hand, passive tension will not use ATP, and it kind of acts the same way as a rubber band does. As long as you're trying to stretch it, the tension will increase in connective tissue. And we'll discuss this better now. So now we will go back to this slide to explain active tension. Now, as I said, active tension is mainly determined by your contractile elements, actin and myosin. And over here, we have our actin and myosin, but we have them squished, okay? This doesn't really happen in real life because in real life, your body is created ideally. And what happens is that your body already maintains something that we call the optimal length. Okay, so this is the optimal length for us, where our actin is not overlapping with each other, and our myosin perfectly overlaps with our actin. Compare it to what's happening in this structure. In this structure, our two actins are actually overlapping each other, and this is going to ruin contraction. Look at how this structure is teamed up. Look at how the myosin is not going to overlap with the actin properly. So when your muscles are squished together, when their length is decreased, they will not have effective contractions. And the amount of contractions or the amount of force they generate will increase incrementally as you are reaching your optimal length. And then when you reach the optimal length, that is going to be the max active force that you can produce, because this is your optimal length. This is the best overlap between your contractile elements that will produce the most effective contraction. And we're already created this way. Our tendons are arranged in a way that will stretch our muscle fibers in the best way possible. Now, what happens when you overstretch your muscles is that look at how the myosin stops overlapping with the actin. And this is pretty bad because this is going to start decreasing the quality of force that you have. And that is perfectly illustrated by this graph. When your muscle is squished over here, you have poor force. As you're increasing the way the myosin and actin overlap, you're increasing the force until you reach your optimal length. And this is how we're usually created. And then what happens is when you overstretch your muscles, you're going to end up just decreasing the quality of force that you're producing again. And at the optimal length, you will have the maximum active force, okay? Now, your passive tension, on the other hand, is different. Yeah, your passive tension, on the other hand, is different, okay? Because the way passive tension works is it's like a rubber band. So imagine you're stretching a rubber band. As long as you're trying to stretch it, there's no optimal length. You're just going to continue increasing the tension as much as you're trying to stretch it. Now, let's take a look at the graph. You are increasing as you're stretching the muscle length. So as you're increasing the muscle length over here, you can see that the force or the passive tension is increasing over here, right? Except that it becomes much higher after you go past the optimal length. So here is the optimal length of the contractile elements. Once you exceed your actin and myosin's ability to stretch, your passive tension is going to skyrocket because essentially your connective tissue is just trying to protect your muscle and it's trying to handle most of the force production because your muscle cannot produce that kind of force in that state. Okay? And the last thing from this graph is that total tension will just be both active tension and passive tension added. And on the graph, it kind of looks like, oh, I think my screen disconnected for a moment. Please give me a moment to reconnect it to each other. Okay, please excuse that delay because there was a technical difficulty, but now we're back, alhamdulillah. So as I was explaining, what the doctor wants you to know from this slide is that the total tension is just the active tension plus the passive tension, so active tension plus passive tension. And the way that looks like on the graph is going to be over here, you can see this wiggly line going like this. That is quite simply the addition of both of the passive tension and the active tension in a normal skeletal muscle. Now, I just included another graph just in case the doctor gets you this other graph, because I do recall getting a question on this in my exam. And essentially, what I'm trying to show you is that the maximum active force at the optimal length on this graph is between point C and D, okay? And if they get you both, then I'd just pick D because, yes, just pick D. Now, let's move on to our second factor, because what I've been explaining so far, remember how I said there are two factors that affect our relationship between the muscle length and the active force it produces? I said there are two factors. The first one is the overlap between thick and thin filaments, which I've been explaining, the overlap between the actin and the myosin and how it changes as your muscle is either shortened or stretched. Now, the calcium sensitivity is mostly relevant to your cardiac muscles, right? And the way this works is that in cardiac muscles, when you stretch them, you're increasing their sensitivity to calcium. So this is a stretched cardiac muscle. This is a normal cardiac muscle, okay? This, even though they both have the same amount of calcium, this stretching is going to cause an increased sensitivity of the cardiac muscle to the calcium. So now, despite them having the same amount of calcium, the stretched muscle will react stronger to this amount of calcium because it's more sensitive to it. And this is just a quality that is in cardiac muscles. Now let's talk about the force and velocity graph. The way this graph works is that the force in this scenario means how much weight your muscle is moving against. How much weight is your body trying to carry? Stuff like that, okay? And the shortening velocity from the name, it's just how fast your muscles shorten. And the relationship is obviously going to be inverse. Imagine you're trying to carry a really, really heavy deadlift. Again, you're not going to carry it very quickly because of how heavy it is. Because of the higher load, you're going to have a slower shortening velocity. And the same is true. If you're trying to carry a paper, it's really light. So you're going to have a very fast shortening velocity. Now let's try to draw this graph in terms of the contractions that I explained to you. Isometric contraction. So isometric contraction, as I explained, you are not undergoing any shortening. You are undergoing contractions, but your muscle is not shortening. What that means is that you are not going to have any velocity. So your velocity is going to be all the way zero. We have velocity over here. You're going to be all the way over here at zero. And as I mentioned earlier in the deadlift example, your body is trying to maximize the force it's producing. Why? Because you're trying to lift a force that you've never carried before, or just a force that you simply can't move. So the point is, what can your body do when it can't create motion? It will try to max out the amount of force it's producing, because maybe then it will be enough to create motion. Okay? Point is, isometric contraction equal maximum force, and velocity will equal to zero, so it's equal to this point on the graph. Now, isotonic contraction, on the other hand, is going to be at any point that you do have velocity, whether it was over here or over here or over here or over here, it doesn't matter. The moment you're able to create movement, the moment you're able to create velocity, that means your muscle fibers are shortening while they're contracting, and that means you have isotonic contraction, and it's going to be anywhere on this point of the graph. Now, you might think, okay, does that mean it's Vmax? It is not Vmax, because Vmax can only be true when you're not working against a load. Because this is a normal XY graph, right? So for you to be able to be Vmax, then you're going to have force or load equals zero. And what did I say about force and load? It's the amount of weight your muscle is working against, or that your muscle is trying to move. So Vmax is only true when there's no afterload. Your muscle is moving itself. It's not moving anything. Okay? So what does Vmax depend on? Vmax depends on the type of muscle you're dealing with. And specifically, it depends on the cross-bridge cycle of that muscle. How fast is it? It depends on how fast is the myosin ATPase, and it depends on how much calcium you have available. So these factors are usually determined by the type of muscle you're dealing with. If you're dealing with a fast twitch muscle, then obviously you have a higher Vmax and a faster myosin ATPase. And if you're dealing with a slow twitch muscle, then obviously slower myosin ATPase and slower Vmax. Now, what the doctor is trying to show you in this slide is very simple. Once again, the nerve comes in the middle of the muscle fiber, and they spread out in both directions. That's one. Two, action potentials happen much, much, much quicker than the actual muscle contraction. The action potential happens between 1 to 2 milliseconds. The muscle contraction takes 50 to 100 times longer, and you could see that in the graph. This is the action potential. Look at how long the muscle contraction takes. Now, the four graphs I'm about to show you, three of them are mainly theoretical. They're just trying to explain an idea. And the fourth one is going to be what your muscles usually do, okay? So, if you were to get a muscle fiber in a lab, like a single muscle fiber in a lab, and you were to stimulate one action potential and allow some time, and then the other action potential, this will create what this graph looks like, okay? These are individual separated contractions that allow the one action potential, then a contraction follows, and then another action potential, and then contraction follows. Point is, you're allowing your muscle to fully relax before you contract again. And again, this is just something that happens in a lab. Your body doesn't work that way. Now, if you increase the amount of taps to around 20 hertz, so tap, 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 what's going to happen is that eventually your muscle is not going to relax and then contract and then relax. What's going to happen is that it's going to contract, and then it's going to contract again, and it's going to contract again, and that will increase the force. Okay? And then what happens over here is that this is a partial fusion. So, okay, the one before was summation, and excuse me, the one before was summation, and that makes sense from the name. Summation means you're adding up. And here you're adding up until you're reaching a new force. But what's happening here is that because of how fast the action potentials are happening, you're going to immediately jump to a high force and then eventually come back down once you're done stimulating the muscle fiber. Now, this is how our muscles actually work, like normal voluntary muscle contractions, okay? They're usually complete tetanus, not tetanus, the thing that I explained earlier, like the toxin. It's just the way that your muscle contracts, okay? Essentially, your body is going to send a ton of action potential signals. So a bunch of nerve signals are going to come to the neuromuscular junction, and then it's going to tell the muscle that you need to like hold a sustained contraction, which is exactly going to happen. You jump to the force that you want, you hold it sustained until you can relax. Now let's talk about what a motor unit is. A motor unit is simply, it's the motor neuron and all the muscle fibers it innervates, okay? Some muscles in your body are going to have only 3 per neuron because of how delicate the movements you want are. Okay? Other parts of your body are going to have one neuron innervating 500 motor fibers, and this is because of how much force you're trying to produce in these muscles. Essentially, what the doctor is showing you here is you have motor neuron A. Motor neuron A is innervating 1, 2, and 3. It's going to make this much force. On the other hand, motor neuron B is going to innervate 1, 2, 3, 4. It's going to make this much force. So the way your body works, obviously, it's not every motor neuron works separately. A bunch of motor neurons innervate all of their fibers together and create this big summed force. That is how your body works. Now, what the doctor was explaining here was he was explaining this ratio. And this ratio is the ratio of motor neurons to the fibers they innervate. In tibialis anterior, which is, I think, a muscle in your leg, obviously you're going to one motor neuron is going to innervate a lot because it's in your leg. Imagine you need to jump, you need to run, you need to make big, forceful movements. So one neuron will innervate 600 fibers. Hand muscles, they're a lot more precise, right? It's not about force, it's about the preciseness of the movement. And you still need a good amount of force. Imagine if you're not able to like unclench and clench your fist. So one neuron will innervate almost 100. Eye muscles, on the other hand, is not about force at all. It's about how delicate and precise the movement can be. So one neuron will innervate 10 fibers. So the question, the force of which of these muscles can be finally regulated? It is the eye muscle. Finally, we're getting into the details of the skeletal muscle types. Type 1 is the slow twitch oxidative muscle that I've been speaking about earlier. These muscles are usually found in your back because they're used for posture. They're also found in your legs for standing and walking. So the whole point of these muscles is that you're trying to do high endurance movements, right? And that means you need to be able to sustain these for long, long periods of time throughout the day. And that means the most important form of energy for these muscles is aerobic metabolism. And that means you're going to use oxygen to undergo oxidative phosphorylation to make ATP. Now, that's why these muscles are going to need high mitochondria, high capillaries, and high myoglobin. Because who's going to carry the oxygen? The myoglobin. Where are they going to get it from? The capillaries. Where is it going to be used in? The mitochondria. So these muscles usually have high mitochondria, high capillaries, and high myoglobin, and the myoglobin makes them red. So that's why they're called red muscles too. And they have slow myosin ATPase activity because we don't care about speed with these muscles. We just want to ensure that we're maintaining the contractions for a long period of time and that our muscles can resist fatigue. Because imagine not being able to support your back after just one day of work. Now, type 2B, not 2A, type 2B is the fast twitch glycolytic muscle, also known as white fibers. And these muscles are usually used for your quick, powerful bursts. So when you're trying to do a quick sprint, when you're trying to jump, you need a quick boost of energy that you don't usually need when you're walking or standing. So these muscles are going to use the anaerobic metabolism and the creatine phosphate, but creatine phosphate is like limited. Anaerobic metabolism isn't really limited, it just has a limitation. So you don't need a lot of mitochondria in these muscles. You don't need a lot of capillaries or myoglobin, and that's why they're white, they're pale, and they have a fast myosin ATPase because you want that fast contraction. And of course, they're going to fatigue quickly. Nobody just sprints the entire day. You just sprint for a while, and then you exhaust that muscle. And then type 2A is just an in-between. So if you want more details on type 2A, you can just read, I think, the chart, and you could see how it's like intermediate in between the two. But because not everything in your body is black and white, of course you're going to have an intermediate in between them. That's all there is to it. It does a little bit of both. And now we are going to move on to the last lecture, lecture number 4, which is smooth muscle contraction. And without further ado, let's begin. So let's begin with the innervation. Smooth muscle can be innervated. Smooth muscle, to make a contraction, can either do it through the innervation of the autonomic nervous system. So the autonomic nervous system will it will send a signal which will cause depolarization, which will trigger an action potential and cause you to contract. Or smooth muscles also have the intrinsic ability to fire spontaneously and contract without even causing an action potential. So they'll just contract on their own. Now, how do we trigger a contraction? We obviously need calcium. And where do we get this calcium from? We get it from two places, from the sarcoplasmic reticulum
and the extracellular compartment, which is like the cardiac muscle, but it's unlike the skeletal muscle. So let's talk about these sources in more details, because there is also a difference between the cardiac muscle and the smooth muscle. In smooth muscles, how do we get it from the extracellular compartment? We don't really have the T-tubule formation that we do in cardiac and skeletal muscles. In smooth muscles, we have 3 channels on the surface of the smooth muscle that open up and allow calcium inside. The first one is the voltage-gated channel. How is it going to open up? By a membrane potential change. So this one is most likely involved when you have depolarization from your autonomic nervous system. It's going to sense that there's depolarization, and it's going to open up, and it's going to allow calcium inside the cell. The second type is ligand-gated channels. And the way these work is that a hormone or a neurotransmitter is going to come and bind to these over here, and it's going to open them up, and then you're going to have calcium rushing inside the cell. And then you have your mechanosensitive channels, and the way these works is because certain parts of your body, like blood vessels, when they get stretched, these mechanosensitive channels will open up, and they will allow for calcium to come in so they can contract back again. So what are your 3 different types of channels found in smooth muscle cells? The 3 different types of channels are going to be voltage-gated channels. These work by depolarization, and then they open. And then we have ligand-gated, either a hormone or a neurotransmitter will come and open them up. And then you have your mechanosensitive channels, which open by stretch. Literally, the cell stretches and it opens. Now let's talk about your cardiac muscles. Cardiac muscles, we've already explained earlier. It depends on the DHP. You have the T-tubule formation because just to jog your memory, cardiac cells and skeletal muscle cells have sarcomeres. They're striated muscles. Smooth muscles don't have that sarcomere formation, so they don't have that T-tubule organization and everything, okay? Cardiac muscles, it will go down the T-tubule, it will depolarize, trigger the DHP to open. Now you have calcium. The calcium will bind to the ryanodine receptor, calcium-induced calcium release. Let's talk about the sarcoplasmic reticulum calcium release, because we said it comes from the extracellular compartment and it comes from the sarcoplasmic reticulum. We already discussed the extracellular compartment. Let's see the sarcoplasmic reticulum. Once again, a neurotransmitter or a hormone will bind to a receptor on the smooth muscle cell membrane. And then this time, it's going to trigger the G protein to activate. The G protein will go and activate something called PLC, which I think is phospholipase C. Yeah, phospholipase C. Phospholipase C will activate, and then it will go and activate IP3. IP3 will go and bind to a receptor on the sarcoplasmic reticulum, and then it will just induce calcium release. So once again, IP3 pathway, hormone, or a neurotransmitter will go bind to G protein. G protein is going to go activate phospholipase C. Phospholipase C will go and produce IP3, and then IP3 will bind to the IP3 receptor on the sarcoplasmic reticulum. Boom, it's opened, and you have calcium release. Now let's talk about the calcium-induced calcium release. It's not really similar to the cardiac cell. There is a difference. So what happens is you will get from one of these 3 channels, you're going to get the calcium, and it's going to enter the cell, and it's going to go and bind to another different receptor from the IP3 one. It will bind to it and cause calcium-induced calcium release. Now, why is this different from the cardiac cell? Because number 1, there's no DHP. That's the number 1 difference. Number 2, the receptor over here is not ryanodine. It's not the ryanodine receptor. We don't have a ryanodine receptor. So we don't have DHP or ryanodine. We just have a calcium-induced channel. Now, remember when I was talking about actin filaments and how the whole concept of tropomyosin covering the myosin binding site on the actin filament was important? The reason why it's important is because that's another difference between smooth muscle cells and skeletal muscles. Skeletal muscles will depend on thin filament regulation to prevent contraction when you're at rest. Okay, so when you're at rest, the tropomyosin is covering the myosin binding site, and it's preventing the myosin head from binding to the actin. Again, myosin is ready to bind to actin. It has the ADP and P. That's all you need for you to bind the myosin head to actin. But the issue is the binding site is hidden by tropomyosin. So this is thin filament regulation, and it's specific to skeletal muscles. Now, smooth muscles, on the other hand, have thick filament regulation. Thick filament is obviously the myosin head, and the way, like, it's the entire myosin molecule. And the way this works is that the myosin in smooth muscles is the same structure as the myosin in skeletal muscles. There is another molecule in smooth muscles called the myosin light chain kinase that needs to phosphorylate the myosin light chain for it to activate the myosin in smooth muscles. So I'm going to say that again. Myosin itself is the same structure between skeletal and smooth muscles. However, in smooth muscles, we have another component, we have another molecule called myosin light chain kinase. Your myosin in smooth muscles is usually inactive, okay? It's usually inactive until this myosin light chain kinase comes and phosphorylates it to activate it. And how does it activate it? How does this activation happen? First of all, the calcium that's been released from the extracellular compartment and from the sarcoplasmic reticulum is going to go and bind to something called calmodulin, okay? Now, this calmodulin is basically going to replace the TNC because there is no TNC in smooth muscle. So calcium binds to calmodulin. This calcium calmodulin complex is going to go and activate the myosin light chain kinase. And then the myosin light chain kinase, using one ATP, will go and enable the myosin to go bind to actin. Okay? So calcium binds to calmodulin. Calcium calmodulin will go activate myosin light chain kinase. Myosin light chain kinase will use one ATP to activate the myosin. And now your myosin can go and bind to actin and perform contractions. Okay? That's essentially what happens there. The cross-bridge cycle in smooth muscles is the exact same as the cross-bridge cycle in skeletal muscles. Just, of course, remember that you need the myosin light chain kinase to phosphorylate the myosin head to keep it active. So if anything happens that unphosphorylates or dephosphorylates the myosin head, it will be inactive and you can't redo the cycle. Aside from that, as long as the myosin light chain stays phosphorylated, cross-bridge cycle will occur in the exact same way that I explained earlier. Now, how do we stop contraction? First of all, the step number one is that you need to have ATP bound to the myosin head, because remember, I said cross-bridge cycling works in the same way. So ATP needs to bind to the myosin head, to detach it from actin. Okay? ATP binds to myosin head, detaches it from actin. Then what happens is that the myosin light chain phosphatase, MLCP, is going to come in. So it's a different molecule than MLCK. MLCP will come and dephosphorylate the myosin light chain. Okay? And once that happens, you are going to stop contraction and you can begin the process of relaxation. I'll just say this one more time. The same step that we have in the skeletal muscle cross-bridge cycle, you're going to need ATP to bind to the myosin head to detach it from actin. That's step number one. Because if this doesn't happen first, you're going to face a problem that I'll get to later. So step one, ATP binds, myosin detaches from actin. Step two, MLCP, which is myosin light chain phosphatase, is going to come and dephosphorylate the light chain, which is going to inactivate it. Now you can begin with relaxation. So let's talk about what would happen if you didn't do it in this order, okay? What would happen if you didn't bind the ATP first? To do that, I'm going to have to introduce phasic and tonic contractions. Phasic contraction is very simple. You're going to have a very normal contraction and then relaxation cycle. So you'll have increased cytoplasmic calcium, and that will cause the cross-bridge cycle. And then after that's over, the calcium will return back to baseline, and you're going to relax. And this is seen in peristalsis. You contract, and then you relax. And then you contract here, and then you relax. And you contract here, and then you relax. Because you're pushing the food down. Okay? Very simple. You contract, you go back to relaxation and to normal. In tonic contractions, you will have the same process of initial spike in calcium, and then calcium calmodulin, and then this will activate MLCK, and this will phosphorylate myosin light chain. The point is, it's going to have the same initial steps as here. You're going to increase your calcium, create a cross-bridge cycle, but you will not return to baseline, okay? What will happen instead is that calcium levels and phosphorylation levels will drop, but they will not return back to baseline the way they do with phasic contractions. I just want to clarify something because I think the slides will make it a bit confusing. I just wrote more steps here But the point still stands in both of them. Like, even in phasic contractions, you're going to undergo the same initial spike of calcium, and calcium will bind to calmodulin. Don't get that confused. This will happen in both phasic and tonic contractions. It happens in all smooth muscle contractions. Okay? The difference between a phasic and a tonic contraction is that in phasic contraction, calcium returns back to normal once you're done. In tonic contractions, calcium and the phosphorylation, both of these do not go back to normal. They just decrease. So they're not as high as they were during the contraction, but they're not back to normal. However, the force of the muscle, or the force produced by the smooth muscle, will remain high. Now, why does this happen? Like, how on earth does your body just randomly do this? It's, of course, it's not random. Nothing is random in your body. What happens is that usually after you're done with contraction, you're supposed to have ATP binding, and then the ATP will release the myosin head from the actin, and this will cause detachment. And then from there, you're going to have MLCP dephosphorylating the head, leading to complete relaxation. In what we call a latch state that happens in tonic contraction, what happens is you will dephosphorylate the myosin light chain before you detach from the actin. So what's going to happen is now you have an inactive myosin head that is stuck, which means you're going to use less ATP because it's stuck. You can't use ATP to free it anymore. And this is going to also help you maintain the same the same force of contraction. Okay? So when you dephosphorylate the myosin head before it's released from the actin, you're going to make it stuck, and the ATP can't free it anymore, and you will maintain the same force of contraction. This way, you can still have contraction for a long period of time without using ATP. And this is specific to smooth muscles. And why is this important? Because, as I said, you want to stay contracted for a long period of time without using ATP. And where is this relevant? In stuff like your sphincters. Your pyloric sphincter is a smooth muscle, and we want it contracted all the time, of course. So it's going to be in a latch state. Now the doctor just put two very simple slides. Over here, you can see that the calcium goes up and then it goes down. In phasic, velocity and the phosphorylation go up. They go back to baseline, and the force of contraction goes up and goes back to baseline. Tonic contractions is different. Calcium goes up, and then it does not go back to normal. It just decreases, and then velocity and, more importantly, phosphorylation goes up, and it does not go back to normal. But contraction force, on the other hand, maintains its level because it's in a latch state. Now, one more thing about phasic and tonic contractions. Phasic contractions are usually single unit contractions. So that means the cells are connected to each other by gap junctions. And what happens is you only need one autonomic neuron. It will trigger the action potential and contraction in these cells. And from there, it can just spread through gap junctions. Okay? And this could be seen a lot in the gastrointestinal tract, specifically during the peristalsis stuff in your esophagus. Now, the tonic contraction, on the other hand, is usually for very precise movements, like in your eyes, So what's going to happen is that there is no gap junctions between the cell. So what's going to happen instead is that it's going to be individually innervating every group of cell. Now we're going to enter the regulation. We're almost done. So now we're going to enter regulation of smooth muscles. In a very general way, whenever you increase MLCK activity, you're going to have more contraction. And whatever you do that will increase MLCP activity, you'll have more relaxation. If you forgot where these two came from, or you just want a refresher, MLCK will phosphorylate the myosin head and it will activate it, meaning it can contract now. And MLCP will dephosphorylate the myosin light chain, which will inactivate the head. And now you'll not be able to contract anymore, which obviously means you're relaxing. Now, what's special about regulation in smooth muscle cells is that they don't only depend on calcium the way cardiac and skeletal muscles do. Because the way we usually regulate and trigger relaxation in cardiac and skeletal muscles is through getting the calcium back where it came from. Now, in smooth muscles, we do that. We do get the calcium back where it came from. And when you decrease the intracellular calcium through the same pathways that we explained before, then you're going to have less calcium calmodulin, and then you're going to have less MLCK, and now you're going to have more MLCP because they exist in a ratio, okay? When you have more MLCK, you have MLCP, and when you have more MLCP, you have less MLCK. So when you decrease calcium, through the same way I explained in the skeletal muscle, and actually more correctly, through the same way I explained in the cardiac muscle, what's going to happen is that you're going to have less calcium and calmodulin, and then that's going to lead to less MLCK activation, which means more MLCP relaxation because they exist in a ratio, and that will lead to relaxation. Now, how do we get the calcium outside of the cell? As I said, you're going to have the same circa pump as you did in the cardiac cell, and then you're going to have the NCX antiporter, the same one from the cardiac cell, but instead of the calcium ATPase in the cardiac cell, you're going to have the HCA antiporter. Okay, so once again, circa same as skeletal and cardiac, NCX same as cardiac, and then HCA is going to be specific for smooth muscles. Now let's talk about the other methods aside from calcium, because I said smooth muscles don't only depend on calcium to relax. They also have other methods. So number one, a neurotransmitter or a hormone is going to bind to a receptor, and this is going to cause increased levels of adenyl cyclase, this molecule over here, and that's going to cause an increase in CAMP. And then when you have increased levels of CAMP, then that's going to activate something called PKA, okay, protein kinase A. Now, pay attention here. Protein kinase A will go and act on MLCK by phosphorylating MLCK. There's a lot of phosphorylation going around here, and it will get confusing, so pay attention. PKA, protein kinase A, phosphorylates MLCK. This will inhibit MLCK. So MLCK phosphorylates the myosin light chain, and this activates the myosin head. However, if PKA, protein kinase A, phosphorylates MLCK, that will actually inhibit MLCK's activity. And this means that if you're decreasing MLCK, what are you increasing? MLCP. What's going to happen? Relaxation. The other pathway is nitric oxide. Nitric oxide is going to go inside the cell through its receptor, and this is going to stimulate guanylyl cyclase. And what this will cause is it's going to increase the levels of cGMP. And because of increased levels of cGMP, you're going to activate protein kinase G, and this will stimulate or increase the activity of MLCP. And because you're increasing the activity of MLCP directly, what's going to happen? You're going to decrease MLCK, and you're favoring relaxation. So nitric oxide is clinical application is that it's usually directly causes vasodilation of it just causes vasodilation, because what are the vessels made of? They're made of smooth muscle cells. Okay. Now let's talk about protein kinase C. Protein kinase C is not actually does the opposite action of the other two that I just mentioned. Some neurotransmitter is going to bind to a receptor, and that's going to cause a long cascade that will trigger protein kinase C, and then protein kinase C will go and inhibit MLCP. And if you're inhibiting MLCP, what are you increasing? MLCK. What will you have? A stronger contraction. So all of these you do need to understand and memorize, but the most important point to take away and to remember is that smooth muscle, you can use calcium to modify it and to cause relaxation, but you can also have other methods to either increase contraction or to induce relaxation. Now, I think we just have a couple of slides with a few extra points. Over here, smooth muscles, they don't have that sarcomere structure, so they're kind of like less organized than skeletal and cardiac muscles. And the way the thin filaments are anchored to the cell, specifically to the cell membrane, is through alpha-actinin, which is found in the Z-band. For more information, refer to your histo lecture. It follows the sliding filament hypothesis. This is just a very fancy way of saying actin and myosin undergoing a cross-bridge cycle. Just a fancy way of saying that. And then finally, the filaments are arranged diagonally, as you can see over here. Diagonal, diagonal, and diagonal. Now let's talk about force length. The force length graph, the same one that we were discussing earlier in skeletal muscles. What you need to know is that it's going to be wider in a smooth muscle cell. Why? Because their filaments are longer. Their thick filaments, their myosin filaments are longer. And what do you need to make this graph, specifically this one? It depends on your actin and myosin filaments. So number one, their filaments are longer, meaning their optimal length is longer. So there you go, wider graph. Second of all, their thin filaments stay connected to the thick filaments even when they're stretched. So the optimal length just lasts for longer. And finally, passive tension is higher in smooth muscles. Why? Because you have more connective tissue. Okay. And I do believe this is our last slide. So smooth muscle velocity in general is just much lower than skeletal muscle velocity, because you just don't need that much. You don't need to have high speed contractions with your smooth muscles. Your body is just not created that way. Okay, so that's why your Vmax is already much lower. If you take a look over here, this is the skeletal muscle and this is smooth muscle. It's much lower naturally. And as I mentioned, that's because your smooth muscles are optimized for sustained energy efficient contractions. And finally, maximum force generation is equal between smooth muscle and skeletal muscles, meaning your smooth muscle may contract slower just normally, but they can generate high forces the same way that your skeletal muscles do. And alhamdulillah, we are finally done with 4 lectures. This actually came with a lot of technical difficulties on my end, but alhamdulillah, we're finally done. If you guys have any questions, please feel free to contact me on my phone or on my email. And all the best. It's your last block. Really try to lock in and to do your best. And have a good day.